Thank you very much, and thank you for the kind invitation to speak. So I'm going to talk about some recent work on understanding universal robustness in pattern formation. And everything that I speak about today is joint work with my colleague Philip Pearce, who's also at UCL. And the ideas that I'm going to talk about today fall under this very broad umbrella of pattern formation. And the specific applications that I'm going to discuss are biological ones. But I do want to emphasize at the outset that the ideas I'm going to present are universal to many types of pattern forming system in general. And I aim to demonstrate why this is the case later in the talk. And while the motivation for this work is in these biological applications, my research interests tend to involve physical problems in applied mathematics more widely. So I don't know much about turbulence or statistical mechanics, but I think there are potentially links between the ideas that I'm going to talk about and the ideas that people in this room are interested in. And so the reason this work is motivated by biological applications is because pattern formation is a very important idea across many areas of biology, particularly when cells interpret external signals, so for example, chemical, mechanical, electrical, and they interpret these input signals to make decisions, or outputs really, based on their surroundings and their neighbours. And so to understand the ideas of this talk, you don't need to know that much about the biology, but it is helpful to know that these ideas are very significant in at least two big areas of biology. And so that's in developmental biology, typically in morphogenesis, which is how tissues and organisms grow into roughly what they're supposed to, and the idea of Turing patterns and positional information in all of this, but also in microbiology, so where people typically care about understanding collective behaviour in communities of microorganisms. So there are specific medical and scientific reasons why people care about these, but from a big picture point of view, this area is kind of important because these types of systems in microbiology are possibly the simplest biological systems that demonstrate emergent complex behaviour. So for this reason, they are widely studied in biophysics. And in all of these biological systems, you're generally acting in a complex environment where the input signals that need to be decoded will vary in space and in time. So for example, in digit formation, you can have a spatial morphogen gradient. And this can also act as a temporal pulse in a developing limb. And it's thought that this tells the cells when and where to form the digits during development. And in bacterial signaling, in microbiology, colonies very often live in environments with unsteady fluid flows. And these can lead to spatio-temporal gradients in the signaling molecules that they use to communicate. So biological systems take in these spatio-temporally varying inputs, and they process them in some way to feed into outputs. And these outputs generally mark decision points in the system, switch-like transitions in the emergent outcome behaviours. But if you have spatio-temporally varying inputs with quite sensitive outputs, you might think that reliability would be an issue. However, biological systems are very widely observed to have very robust functionalities, robust outputs. So how is it that they are able to do this? And this question of robustness is one of the big questions in biology. It spans across many different contexts. It's been investigated in many different systems. And so when you talk about robustness, you kind of have to say robustness in what and with respect to what. So sometimes robustness means with respect to parameter sensitivity, i.e. between different individuals. We've been developing a framework to try and understand aspects of robustness in a different context. And that's with respect to spatio-temporal variations in input signals, specifically over timescales faster than growth. And the very simple robustness question that we started thinking about was, when do spatio-temporal variations in the input of a biological system get filtered out of the output of a system? And when do they get reflected in the output? So when does the system ignore them, gain robustness to them, And when does the system incorporate these into its decision making? So to get a handle on this question, we just took a simple model from the digit formation literature where these pattern-forming systems are well parameterized. And there's a wide range of complexity that you can add to these models. We took a very, very simple model just to see if anything interesting could happen. In this simple model, the entire hierarchy of morphogens, which essentially are just signaling chemicals that induce cellular responses, the entire hierarchy of these is relegated down to 2 interacting chemicals to solve for. And the precise details aren't too important, but you can basically just think about this essentially as an activator A and an inhibitor I. So you just get this activator-inhibitor type system shown here. And in this model, there is a prescribed input pre-pattern morphogen that feeds into the system. This is an input to the system quantified through this spatio-temporally prescribed function f of xt. So more broadly, in a complex system, you might have to solve for this function, but here we prescribe it because 1, it's much simpler to do that, and 2, it doesn't affect the main ideas of this talk. And this function f is spatially heterogeneous in the steady system that we take it from in the literature. And we basically say, what if we also vary it temporally, kind of a proxy for complex environmental effects? When would we expect these variations in the input to get filtered out of the output, for the system to sort of treat them as noise and ignore them? And when would we expect them to get reflected in the output, for the system to kind of treat them as signal and take them into account? So we can just solve this very simple system numerically. The prescribed input is at the top, and 2 representative outputs are shown at the bottom. And the only difference for the simulations of the outputs at the bottom is that on the left it's a slow input variation, on the right it's a fast input variation. And the timescale in all of these has been normalized to the variation timescale in all of these videos. You can see that you get a robustness effect. The output patterning in the bottom right, on the bottom left is reflecting the input variation, but on the bottom right it's filtering out this input variation at the top. So why is this happening? Well, the standard thing to do at this point is to look at the natural timescales in the system and see how the variation timescale compares to these. So broadly, you'd maybe only expect qualitative changes in behavior when the variation timescale crosses a natural timescale in the system. So if the variation timescale is much faster than natural timescales, then you probably don't expect the variation to get reflected in the output because the system just can't respond to those changes. If it's much slower, sort of the quasi-steady limit, then you probably do expect the variation to get reflected in the output because the system can respond over these timescales. If it's somewhere in the middle, well, you might expect the reaction terms to reflect and diffusion terms not to, but the key point is that in the middle, you don't expect any qualitative change, any qualitative difference in the output for a variation timescale between 1 minute and 20 hours, because you're not crossing any natural timescales in the problem. But this is kind of strange from a biological robustness point of view, because physiological, which is growth in this system, physiological timescales in this system are around 10 to 24 hours. So can it really be the case that any variation with a timescale between, say, 1 minute and 10 hours should behave in the same qualitative manner? Biologically, this is definitely not what happens. Biological systems seem to be robust to and to filter out variations that are a few hours shorter than physiological timescales. So do we need to add more biology into our model, this very simple model, in order to understand this experimentally observed robustness in these types of systems? Well, we can sort of check this in our model just by simulating variations over these intermediate timescales between these natural timescales of reaction and diffusion, just to see what happens. And that's what you see here. So for 30-minute oscillations on the mid-left, the patterning seems to be fairly robust to the input changes. The pattern is largely maintained. But for 10-hour oscillations on the right, the pattern is massively affected by the input variation. The input variation is completely reflected in the output. So we do somehow seem to be getting robustness at the appropriate biological timescales here, but we have not added in any extra biology into this very, very simple model. So why is this happening? It turns out that part of the answer is that there is a bifurcation in the system, or a phase transition, depending on whether you're a mathematician or a physicist. But this isn't something special about this digit formation system. This is a feature of many pattern-forming systems in biology and beyond. It's certainly true for any Turing pattern system, because the pattern manifests through a Turing bifurcation. But it's also true for many non-Turing patterns that are studied, including positional information systems in biology. Very often, patterns in these systems manifest through a change in stability of some process. Some self-accelerating interaction takes over from a self-decaying interaction. And this means that many types of biological pattern manifest mathematically through bifurcations or phase transitions by a sudden change in output for a small change in input. So in biology, you often, but not always, see pitchfork bifurcations for activator inhibitor type systems and transcritical bifurcations for self-activator systems. And the reason that bifurcations are important here is that timescales slow down near bifurcations. This is, of course, a classic result from nonlinear dynamics. And it basically happens because the stability is changing at a bifurcation. Mathematically, this is generally because your bifurcation parameter mu gets small close to the bifurcation. And so just comparing the two important underlying terms here, you can see that the classic slow relaxation timescale is 1 over mu, essentially saying that the timescale gets very large close when mu is small, i.e. close to the bifurcation. We saw that this morning as well. So is that what is happening here? Well, if you apply this to the digit formation example and calculate the slow relaxation timescale you get from that type of analysis, you get a prediction of the critical robustness timescale of around 10 minutes. But if we look at our timescale schematic, 10 minutes is still too quick to explain this observed intermediate robustness. So this classic result of a slow relaxation timescale near a bifurcation is not enough to explain what is going on here in this system. In fact, to understand this sort of apparent robustness, we need to be more careful in how we evaluate the effect of the bifurcation, this phase transition. And the key thing is that the effective position of the bifurcation is moving around. So the system is essentially continually crossing the bifurcation dynamically. And it's this dynamic crossing of the bifurcation that is causing a critical slowing down effect over non-standard timescales. So this is a sort of well-known effect. It's called the delayed bifurcation effect. It's been shown to be fundamental for other physical systems over the years. And it turns out that it's this moving or delayed bifurcation effect that generates the appropriate robustness timescales observed here. That is, that there are slow timescales, slower than classic slow bifurcation timescales. And these additional slow timescales are generated by the movement of the bifurcation within the system. And as we'll see, these are what cause the emergent robustness observed at the intermediate timescale. So to give you a sort of intuitive idea of why this happens and the reason that it's fundamentally different to classic bifurcation slowing, the key idea is that your effective eigenvalue is now crossing zero dynamically. So for a non-degenerate crossing of the bifurcation, it's going to look close to linear if you zoom in enough. And then we can say that a given crossing occurs with some gradient or rates lambda. And then exploiting this near the dynamic delayed bifurcation crossing, the system basically looks like this. And then just by a scaling analysis of these two terms, of these underlying terms here, you can see that you get a critical slowing timescale of 1 over the square root of lambda. So what this means is that the actual slow timescale here depends on lambda, which means that it depends on the dynamics of the bifurcation motion, essentially on how quickly you pass through the bifurcation. So this is fundamentally different to the classic bifurcation slowing, which only depends on how close you are to the bifurcation, not on how quickly you are moving through it. And this is kind of really important for the whole idea of the talk, so I really want to flag this idea. This effect is not just a mathematical curiosity. This effect cannot be captured by a classic equilibrium analysis. And if you do not take this inherently dynamic effect into account, you get things wrong and you cannot properly explain the emergent robustness observed in this system. So for our problem, and for these types of system more generally, the technical way that we can analyse them involves scaling in near the moving bifurcation and deriving the appropriate modified normal form through a modified weakly nonlinear analysis that systematically takes the dynamics and spatial heterogeneity into account. So because the digit formation example involves some complications that I'll get into in a moment, it's easiest to first see how this works on a toy example. And this is really the simplest toy example we could think of that captures all of the important effects. And many classes of biological pattern forming system will collapse down to the same canonical normal forms that I'm going to derive from this toy problem. So essentially this toy problem is a dimensionless reaction diffusion problem where this k of xt is the spatio-temporally varying prescribed input to the system. And this can be positive or negative. All the other terms here have their usual reaction diffusion meaning and are positive. So k being positive means sort of a net self-activation. K being negative means a net self-decay. And this means that k equals 0 defines the position of the bifurcation here. And the three parameters that I note at the bottom are small. This is just so that the system looks like it should be quasi-steady and locally forced by the reaction terms for ease of exposition. And at this point, yeah, I'll make no further assumptions on their relative size. And then the last important thing to note is this exponent n in the nonlinear saturation term at the end. This tells you the type of bifurcation that you have. Specifically, we're going to consider two cases. N equals 2 is a transcritical bifurcation. N equals 3 is a supercritical pitchfork bifurcation. We've done the analysis for both, but if you prefer definiteness, I suggest choosing N equals 3. This corresponds to the specific Turing bifurcation in the digit formation example that I'll get back to, but also in many activator inhibitor systems more generally. But the key thing, yeah, go ahead.
What's little a?
It's sort of base production, a base production of the chemical in question of this reaction.
Okay.
I'm sorry, can you say that more loudly?
It's a base production of the chemical of interest. But the key thing from what I'm about to do is that the universality of bifurcation means that any reaction diffusion pattern forming system that shares the same bifurcation type as this system, which is pitchfork or transcritical, will have the same robustness properties as we're going to learn from this system. And the way to derive these is to scale into this dynamic inner region around the moving bifurcation, essentially around the transition in solution behavior. And here the moving position of the bifurcation is defined implicitly at the point at which k is equal to 0, which, as I said, is where you transition from self-activating to self-decaying in the quasi-steady problem. So to find the appropriate scalings, you can look for inner scalings that capture as much of the biology as is mathematically consistent. So in the language of matched asymptotic expansions, which is essentially what we're doing here, this is essentially looking for the distinguished asymptotic limit, i.e. the least degenerate limit. And then any specific problems, including the one for the digit formation example that I'm going to get back to, will end up being special cases of the full unifying general problem that we're just about to derive. And basically, by balancing these last three terms, you get the appropriate scalings given here. Then, by imposing these scalings and zooming into this moving inner region, at leading order you get this generic inner problem, essentially a modified normal form, that captures the dynamic crossing of the bifurcation. So one slight caveat on this slide is that I've implicitly assumed that the bifurcation is moving monotonically. You do need to be much more careful near turning points, but I will address this later, so for now you can basically ignore that restriction. But essentially this modified normal form involves two parameter groupings. This is lambda and chi. And this lambda term corresponds to diffusive effects. This chi term corresponds to moving bifurcation effects. The +1 to the base production, the zy to local spatial effects, and the -y to the end to saturation effects. And the universality of the bifurcations means that this is the general unifying form for transcritical or supercritical pitchfork bifurcations in reaction diffusion pattern forming systems in the presence of spatio-temporal input variations. And specific biological models like the digit formation example will be special cases of this, and I'll show you that in a moment.
And just kx is the derivative of k with
The
respect to
partial derivative with respect to x, yeah. So that's why it's the local gradient.
Is it guaranteed not to be zero?
Pardon?
Is it guaranteed to be non-zero?
So that's the case when you have turning points, and I'll get to that in a moment. But for now, we're just assuming it's monotonic. But when you're completely right that when it is zero, you need to be more careful in the scalings. But I'll get to that in a moment. And because this is the normal form for these types of bifurcation, we analysed it exhaustively to characterise and quantify all the types of behaviour that are possible in terms of the effective parameters lambda and chi. So I'm not going to go into the technical details, but this diagram summarises the results of that analysis. Every point on this figure represents a solution of the normal form for a different value of lambda and chi. Essentially, it's characterising a phase diagram with all the key limits and the special cases of the normal form. So a general spatio-temporal variation in the input will be a trajectory on this phase diagram. All the technical details are in the paper if you're interested, but essentially the point of me showing you this is that all possible behaviours of reaction diffusion systems with pitchfork or transcritical bifurcations are characterised by this phase diagram. So briefly, in region A, the transition is quasi-steady. It's basically what you would expect if you ignored all of these emergent effects, and there are certain limits in which that is the appropriate thing to do. Region B is where you get this critical slowing down effects, and therefore the robustness effects in the problem. And region C is where you get diffusive enhancement effects, and where these become important. I'll discuss region B in a little bit more detail, and that's because it's the region where you get the delaying behaviour. So it's the most important region for understanding the emergent robustness of these systems. And basically in this region defined through these scalings here, diffusive effects are not that important and chi is large. And so we can analyse what happens in this region to understand that initial observation, the unusually prolonged persistence of the patterned state when you expect it to disappear. And so looking at the transition from patterned to unpatterned means the case where chi is positive. And the case where the pattern forms through a pitchfork bifurcation, which is the case for the digit formation example, and often for other Turing bifurcations, means the case where n is equal to 3. And it's precisely this sort of dynamic crossing of the bifurcation from the pattern to the unpatterned state, this dynamic change in stability that is causing the pattern to persist over these longer timescales, causing the robustness. So to show and quantify this, you can do some asymptotic analysis, essentially to solve for the concentration everywhere within this transition region. And I'll skip over the details of this here, but a key point of interest for the analysis is that it involves exponential matching within logarithmically scaled transition regions. So you do see what is essentially a super exponential growth in the transition region. And so personally, I think it's kind of interesting in its own right, but perhaps less important for the big picture here. But the key result for our purposes is that the concentration in the green region, because this is going to allow us to quantify and understand what is going on in terms of robustness. And basically, this asymptotic result is particularly helpful because we can invert it to get an analytic result for the dynamic position of the transition, essentially the position at which y is equal to 1 here. So for the quasi-steady problem, the position of the transition is when z is equal to 0, because nothing unusual is happening. But from these asymptotic results, this is not the case in region B. Here, it occurs for a non-zero value of z, actually an asymptotically large value of z within this inner region. So this gives us a quantitative result, not just a scaling for the persistence length. And that's that it's the square root of chi log chi over 2, where chi represents the effect of the spatio-temporal variation in the system, essentially the moving bifurcation effect. And it's exactly this persistence length effect, this non-zero dynamic position of the transition, that is causing the emergent robustness in these pattern-forming systems. It's the continual dynamic crossing of the bifurcations that is generating emergent and non-trivial slower timescales. And so the overall system patterning becomes much slower to react to spatio-temporal variations. So what this means is that the biological system can pass into regimes that should cause depatterning from an equilibrium analysis, but without actually depatterning. And they can do this over much longer timescales than you would expect. And this is exactly what is happening in this digit formation example. Okay, so that's kind of the main mechanism that drives the emergent robustness. But one final thing before we can use these results generically to understand specific examples is in exactly that point, dealing with non-monotonic moving bifurcations. So this is important for any case where the spatio-temporal variation moves back and forth, which is certainly the case here, but really any case with a variation around some mean position. So the reason that these need to be treated carefully is exactly because this motion is non-monotonic. It contains turning points. So you still get critical slowing down in these scenarios, but you just need to be a bit more careful in the analysis. And the reason for this is essentially due to how we derive this normal form. So the z in front of this y, in front of this linear term, represents exactly the linearization of the dynamic crossing of the bifurcation. But near a turning point, that linearization is 0, it's sort of degenerate in some sense. And so near a turning point, you just need to be a bit more careful on the scalings and properly incorporate the higher order terms. Basically, this just means that you need to treat turning points slightly differently because otherwise you get the wrong scalings. And to cut a long story short, in region B, which is where these delaying effects are the most important, the key reduced ODE you end up with has this leading order form where F accounts for the specific kind of turning point. So this reduces to the linear case the linear crossing case that I just discussed when F is equal to minus T. But this more general formulation allows us to tackle turning points when the bifurcation crossing is not well approximated by a linear function. But again, you can solve this general nonlinear ODE exactly, now getting this solution in terms of this general function f. So now we can understand what happens for all kinds of bifurcation motion. And specifically, we can now understand why it is that these patterns persist. And so we can understand the emergent robustness of pattern formation for any pattern-forming reaction diffusion system where the patterns manifest through supercritical pitchfork bifurcations. Also transcritical, but the details of that are in the paper. All you have to do is understand how your system reduces to these modified normal forms in the presence of these spatio-temporal variations. And then you can just apply these robustness effects directly to your system. So, given that all of that analysis, let's now reap the benefits and apply these results to the digit formation example that I started with to see what sort of physical insight that we can get into the emergent robustness of that system. So as I mentioned before, the digit formation example involves a slight complication, which is that the inner analysis requires including two inner regions at the same time, basically for the wavelength and the amplitude variations, essentially via a modified weakly nonlinear analysis for a system of PDEs. But this just follows the same broad steps as a multiple scales analysis for a system of PDEs, and it just means transforming your system appropriately. And again, I'll just give you a high-level overview of how this works. The technical details are all in the paper if you're interested. But the analysis starts by transforming into both of these inner regions at once in the standard multiple scales way. There's nothing unusual about this at this point. And then your leading order analysis is basically the same as a linear stability analysis to find the onset of instability. And so this gives you a result for the critical wavelength of the patterns at this onset of instability, which is this Kc here in terms of the system parameters. But importantly, the linear stability analysis cannot give you information about the emergent robustness. It's inherently a nonlinear effect, as we might expect. Mathematically, what this means is that your leading order solution from the linear stability analysis here in the red box is defined in terms of an as of yet unknown amplitude function, which is y of sigma t, where sigma is this intermediate amplitude variation length scale, t is time, and the capital X in the cos is your fast pattern length scale. So to understand the emergent robustness, we essentially need to go to higher order terms and explicitly account for the spatio-temporal variation in the system. So as is normal with a weakly nonlinear analysis, to derive a governing equation for y, we go to these higher orders and derive a solvability condition, and here you have to go to second order correction, which is not unusual for a pitchfork bifurcation. But this epsilon to the 2/3 order is where the spatio-temporal variation comes in to the problem. And then if you derive the higher order problem, you can apply an appropriate solvability condition, just being careful about the fact that it's a system, just applying the Fredholm alternative theorem. But essentially what that means is that you can derive this governing equation for y, which is essentially a governing equation for the amplitude of the patterns in the presence of spatio-temporal variations in input. And we can see that the motion of the bifurcation comes into this y sigma term where there's an s dot in front of it in this coefficient. And the spatio-temporal variation comes in terms of this linear term with a df by dx times the sigma term over there. But most importantly, this governing equation is a special case of the unifying normal form that we saw and analysed previously, but now in terms of the specific digit formation system parameters. So what that means is that we can use the parameter landscapes that we characterised comprehensively to understand the robustness implications for this specific biological system. So let's use this parameter landscape in the top left to understand the implications of this analysis for biological parameter values from the literature. You find that you sit in region C for changes over physiological timescales, which agrees qualitatively with experimental observations. And then you can ask, what if I reduce the timescale of my oscillation? When do I enter region B and start to see these robustness effects? And if you put in the experimental parameter values for the steady analysis, you get a prediction for the dynamic robustness of around 5 hours. So this result completely explains the emergent robustness in the mathematical model of the system. But we think, perhaps more importantly, it gives an intriguing and potentially quite simple explanation for general biological observations of robust functionality. That it's the intrinsic properties of pattern formation, the bifurcations from which they emerge, that are giving you robustness for free over non-standard timescales. And for this biological system, it's a few hours shorter than physiological timescales, which agrees qualitatively with what people see. So that was the general idea applied to a developmental biology example. But as I mentioned at the start, the idea of emergent robustness in pattern formation is also important in other areas of biology. So we can use this same mathematical framework to understand emergent robustness in a microbiology context, in collective bacterial decision-making. And this might seem very unrelated, but bacterial signaling is activated via a transition. And so mathematically, it's controlled by a bifurcation. In this system, we'll show that it's a transcritical bifurcation. So this is going to generate an emergent robustness over a certain time scale, and we can quantify and understand this using our framework. So before I get into this, why are bacteria important to study? Why is microbiology a big research area? Well, at a very high level, bacterial self-organisation is one of the simplest biological examples of emergent complex behaviour. So for this reason, these types of systems are widely studied in biophysics. But there are also specific biological reasons. So bacteria are very important to humans. They're vital for human life, but they can also make us very sick. So the idea is that if we can understand how they enable their synchronized behavior in these complex environments in which they live, hopefully we can understand how to disrupt or control it. And the broad way that they are able to act as a collective is by communicating with each other. And so bacteria communicate through a process known as quorum sensing. Each bacterium produces, releases, and detects these diffusible molecules that are called autoinducers that can cross the cell membrane. And the detection of the local autoinducer concentration allows a single bacterium to sense if there are lots of bacteria nearby. And so this often, but not always, involves a positive feedback process. Essentially, when bacteria detect these molecules, often they will start to produce more of them. And biologically, the onset of quorum sensing activation is observed through a sudden large increase, a switch on of autoinducer in the system. Mathematically, this sounds like a phase transition, and mathematically this will often arise through a bifurcation. And the key robustness question that we looked at was, how can this mechanism deal with the fluctuating flow environments that bacteria live in? So flow removes autoinducers from the system because the autoinducers travel through the environment. So how is it that bacteria can demonstrate robustness in their response to unsteady changes in the flow? How is it that they can ignore some variations in the signal but respond to others? So to answer this question, we just looked at a multiscale model involving a cell layer consisting of bacterial cells embedded within an extracellular matrix, and above this there is a fluid flowing. And within each bacterium, we just include an archetypal genetic circuitry for their positive feedback quorum sensing, which is called it's a classic LUX-IR system, if you know about these sorts of things. So I'm not going to put the full equations up, but essentially it's two coupled regions solving for the fluid flow in the top region and tracking 5 chemical concentrations in total. And if you solve the mathematical system, just in the steady case for now, the numerical results predict this switch-like response that is observed experimentally, shown here as a function of the external shear rate. So if the shear rate drops below a certain amount, stuff is removed from the system at a much lower rate, then you see this sudden switch-on behaviour. Mathematically, this is caused by a transcritical bifurcation in the system. The y-axis here is a log axis, so the change in behaviour across this is quite significant. And this means that the framework that we've just seen can now be used to understand questions about biological robustness in this system, because this sudden switch in the output behaviour is governed mathematically through a bifurcation. So given this, we can think about that key question in trying to understand robustness to fluctuating flow conditions. And to sort of frame that question a bit more explicitly now, essentially, if steady activation occurs when the external shear rate falls below this critical value, what's to stop erroneous activations for fast incursions in and out of this region? Biologically, these systems are fairly robust to these unsteady environmental fluctuations, but can we explain why using our model, or do we need to add in more biology to understand the observed robustness? Well, we can just test whether this can be observed in the model numerically. We just need to oscillate the external imposed shear rate in and out of the activation zone. And so this is a simulation of the autoinducer response in oscillating flow conditions. At the top, you're seeing the fluctuating input of the external shear rate, and at the bottom, you're seeing the output, which is the autoinducer concentration for 2 different oscillation rates. And again, I've normalized to the oscillation timescale in each case. It's a slow oscillation in the bottom left, a fast oscillation in the bottom right. And you can see that the slow oscillations respond kind of as you'd expect, clearly moving in and out of activation. But the fast oscillations don't have this strong response. They only change by a small amount, and they appear to be locked into the inactivated state. So the dynamic effects are clearly important here, but what is the critical robustness timescale, and how does it relate to the natural timescales? So this figure summarizes those simulations, but for a whole range of activation times, which basically means different oscillation periods. And you can see that there's a critical activation time in this system, and you have to spend longer than this in the activation zone to actually experience activation in a fluctuating flow. And importantly, this critical activation time is much longer than any of the natural timescales in this problem. Vertical diffusion here is a fraction of a second because the cell layer is very thin, and reaction timescales here are in the order of minutes. But this critical activation timescale appears to be several hours. So what is causing this? It's exactly the same reason I spoke about in the first application. The biological onset of quorum sensing activation manifests through a bifurcation in the mathematical system. And the dynamic crossing of the bifurcation with the fluctuating flow causes a critical slowing down over non-standard timescales. So we can understand and quantify this by performing another modified weakly nonlinear analysis. And if we do that analysis, tidying up the variable scalings, we end up with this scaled amplitude equation for the concentration around the transition. And this is a sublimit of that normal form I previously showed you, but now for n equals 2, which means a transcritical bifurcation. So we're now in a position to understand the context of these results using that parameter landscape that I showed you previously. And so, even though this is a completely different biological system, the same framework as before allows us to find a similar robustness conclusion. So for changes over physiological timescales, we again find ourselves sitting in region C. Even though the natural timescales here are much shorter than the physiological timescales, you find that you get an emergent robustness timescale of around 3 to 15 hours, depending on the specific parameter values that you use, which again is just a few hours shorter than the physiological timescales. And again, it's the intrinsic properties of pattern formation, the transitions, the bifurcations from which they emerge that are giving you robustness for free over these non-standard timescales. And for these systems, that's a few hours shorter than the physiological timescales. So there are some interesting differences between the systems that arise from doing the analysis. So for example, the diffusive enhancement effect in region C over physiological timescales is much stronger in the quorum sensing example. So what that means is that all of the colony activates at once. And this kind of makes sense biologically because in the digit formation example, you kind of want spatial effects to be confined because you want your digits to form in a specific location. But in a bacterial colony, you want the entire colony to make the same decision. So it's quite nice that this is reflected in the model. And so, more broadly, spatio-temporal variations for any reaction diffusion pattern forming systems with these types of bifurcation can now be understood just by analysing and understanding their phase trajectories on this phase diagram that we've characterised for their modified normal forms. And features of the trajectories, like if and when they cross the boundaries between regions or how deep they go into each region, will give you fundamental physical insight into the emergent robustness of these systems. So this is particularly relevant for biological pattern formation and biological signalling, because in these types of system, the signal is very often a transition in the system that's triggered by a change in stability of some mechanism in the process. But it's definitely not restricted to biology because it's a bifurcation, so there's a universality associated with that. Essentially, these ideas will hold for general reaction diffusion pattern forming systems where the pattern manifests via a bifurcation, and for these specific quantitative results for pitchfork or transcritical bifurcations. So just to summarise and conclude, patterns often manifest through bifurcations mathematically because bifurcations codify sudden changes in output. And we've seen how these intrinsic properties of pattern formation, the bifurcations from which they emerge, give you spatio-temporal robustness for free over non-standard timescales. So for the two biological systems that I investigated here, this was a few hours shorter than physiological timescales. But these results can be applied directly to any system that exhibits these types of bifurcation, not just in biology. So the technical details are all in the papers if you're interested. And just to reiterate, all of this work is joint work in collaboration with my colleague Philip Pearce. And I'd be very happy to chat if you have a system with bifurcations and you're interested in understanding its robustness to spatio-temporal variations. But thank you very much for your attention, and can I take any questions?
So I might have missed something very basic, but I kept waiting to see this, the chi of t have a trigonometric behavior. You had to have a linear behavior in time, and then I thought, well, but then it gets reversed, so you're going to patch it together with another one that goes the other way. Am I mistaken? Isn't that?
So I didn't.
It's just one way, right?
So I didn't write down the specific form of chi of t for this problem, but the first twig.
Lambda t minus tc, right? It was linear in time. Am I not?
Yeah, so that was for the first. That's for a monotonically moving bifurcation. For that problem, you're correct. That was just for a linear crossing. But then the second, the sort of caveat that I mentioned about the turning points in the system, then that's where you have these turning points. And so you're completely right that when you have a trigonometric form for that, which is exactly the case that we had sort of behind the scenes in all of this, then you do need to take into account that turning point, and I did take that into account when it came to calculating the results, but I didn't show the details of that. You are correct, I didn't show the details of that.
I'll ask a question. So, yeah, I'd like to discuss the wave number. So, in the first part, when you did the analysis of your tooth activator inhibitor system, the wave number appeared was still Kc. And is there a reason, was there an assumption as to why it remained at Kc?
So, for this problem, well, basically because it's all a local analysis, because it's essentially a second order transition, because everything is local, it means that because you're zooming into this inner region, you essentially only care about what's happening at the position of instability. So that's why the Kc here is the same everywhere, because for this system.
But wait, just remind me, isn't the, generically the wave number of maximum growth linear in the distance from?
But that's away from the transition, right? The wave number of maximum growth is away from the transition. Whereas this is, when does the system first become unstable? And you're always locally in that point. So these results work for these second order transitions really well, because it's a continuous phase transition, so you only really care about the local behavior of these. There are some things you can do for discontinuous phase transitions, but in general they're much more difficult because you're away from this sort of stable manifold of the system, and you have to take into account exponentially growing terms and things like that in order to make any progress.
Yeah, sorry. So in the interest of
Yeah,
time,
of course.
we'll move on to our next speaker. Thank you very much for allowing me to